Bismillahirrahmanirrahim. Assalamu alaikum. So in the last lecture we started with this topic of membrane separation. Actually from the last week we started with this topic, and in the last lectures I have discussed about membrane material. So if you remember, these were our objectives of that lecture, in which we stated that we have to discuss something about membrane material, that membranes are synthesized of different material, what are those material, and then what are the different types of membrane, and later we said that we will switch towards membrane modules and synthesis. So if you remember that we have discussed in detail all this stuff of membrane material and their types, and then we have to start with membrane modules and synthesis. But before I start membrane modules and synthesis, we will quickly go through the stuff which we have already discussed. So as you remember that we said before 1930 the materials which were used for membrane, they were natural polymer, and after 1930 due to synthesis of various material in the industries, so they are being used for synthesizing various membranes. And we discussed that most of the time polymers are used for membrane synthesis. Even naturally polymer were being used, they were cellulose and rubber, and later on with the development of industries, even again synthetic polymers were being used, and there were different types of polymers, like some are linear, branched, and then some are cross-linked polymers. So when you look at the classification of these polymers, they are classified into thermoplastic and thermosetting polymer. Thermoplastic polymer were those polymer which can be softened with the help of temperature, and you can increase their temperature, and finally you can actually melt them, you can just break them into their monomers, and they can be reused. But thermosetting polymers are those which cannot be broken down, they cannot be melted with the help of temperature, because when you increase their temperature they will at the end decompose and they can't be used again. So then we said that polymer are actually, when you look at the polymer, polymers are long chain of monomers connected together. So they are arranged either in the form of amorphous or crystalline polymers. And amorphous mean they don't have any specific shape, and usually they look like the glass. From here we said that you can call them sometime as even glossy polymer, but they are amorphous, and they lack crystalline structure, proper like arranging of chains. But there are crystalline polymer, they have properly arranged chains, and they are usually opaque, and they will have crystalline structure. In this diagram you can see these portion indicates this is amorphous polymer, and while other these indicate they are like crystalline regions. When you look at this classification, among this that for amorphous polymer we can name them either as glossy polymer or rubbery polymer. There was one temperature that was called glass transition temperature, and when some polymer, that is amorphous polymer, that has temperature, or it is existing at the temperature that is less than Tg, glass transition temperature, then that will be called as glossy polymer. But if you just raise its temperature and you go beyond or above this Tg, the polymer becomes rubbery. And we discussed this, that when polymer is rubbery, actually chains can move, and it allows more diffusion of the permeate, while in glossy polymer definitely chains are more packed, and that's a bit dense as compared to this rubbery structure, so their diffusion will be low. And then when you look at the other side, which was actually crystalline polymer, so we said that they have some property which is called melting temperature, because when you increase their temperature you can reach to a point where this will melt, and when polymer will melt, this can be reused for again synthesizing any product. But this is valid for only thermoplastic polymer which are in crystalline form. If they are thermosetting polymer, they can't be melted, because they will decompose when you will increase their temperature. When you look at the polymers available, they are usually somehow amorphous as well as crystalline. That's not like pure amorphous or pure. Most of the polymers which are available, they contain some portion of amorphous and some can be like crystalline. So when some polymer is a mixture of amorphous and crystalline arrangement, then this will have both Tg and Tm. Tm, the melting temperature, that is property of crystalline polymer, and Tg, that is property of amorphous polymer. Then we have looked at this stuff, and from here you can see there are various polymer they are used and for synthesizing the membranes, and you can see their properties like glass transition temperature or melting temperature are given, and they have various range. So when you look at the permeation through the membrane, we want such membrane because here are now different material. Which material we should select? Actually, we should select such material which is good for our separation. Actually, it provide high permeance of the one component of the feed and overall high permeance ratio. I have talked about this in detail in last lecture. If you can just check what does this mean. And then we looked at this, that this permeance is actually analogous to the something called mass transfer coefficient, and if you look at its equations, that molar flux through the membrane is equal to permeance multiplied by driving force, and this permeance is actually ratio of permeability over membrane thickness. So we talked about this, that how membrane thickness or permeability will affect the separation through the membrane. And then membranes are actually classified into two categories, dense membrane and porous membrane. Dense membranes are also named as like non-porous membrane. They usually don't have any pores, or if there are pores, they are very small, like few angstrom in diameter. And these because most of the time they are non-porous membranes, so here separation didn't take place based on size, it will take place based on various properties. Actually here such material which can diffuse through the membrane can easily be separated from the other material which can't diffuse, and with the help of these membrane, even those material or those feed in which there are more than one component and they are of somehow similar size, they can be separated. How? Because here you can separate them not based on the size but based on other properties of diffusion through membrane material. Through these material mainly this diffusion takes place along the paths of like chains, that your component which can diffuse through the membrane will pass through the polymeric chains and will leave on the other end, and other can't pass through that. Then when you look at the porous membrane, they are also named as microporous membrane, and they were categorized into three types, so ultrafiltration, micro and nanofiltration, and we have seen that the difference between these three is actually based on their pore size difference. And because they have different pores, so here actually separation takes place based on size. They are good for those separation in which you have some smaller and larger molecules, because the pores are of different size and they can help you to go for this kind of separation. They have usually high high selectivity here can be achieved when molecules are different in size, like some are smaller, some are higher. Then you can highly selectivity mean actually the membrane selectivity, that membrane should allow one of the component to pass through and other should stop it. So in these membrane, if you look at the pore define actually that how much will be the flux and rejection. Actually membrane material didn't define the flux and rejection, but when you look at the dense membrane, in those membrane actually your material of the membrane define that what will be flux and how much will be the rejection through the membrane. And then we said that because these polymeric membrane are usually applicable, or they can be used for low temperature like up to 200 degrees C, and they also should be used where the your chemical which you are separating should not react with the membrane. So in all those cases where temperature is high and there is a chance of reaction of your feed with membrane material, then you go for some inorganic material. And in our inorganic material membrane, so they were formed of different material, alumina, there is glass membrane, there are ceramic based membrane, zirconia and all other. So up to here we have discussed yesterday, in which finally we stopped here, that in a broad classification you can look that membranes like have symmetrical and asymmetrical types. Actually symmetrical membranes are those membranes which are symmetric throughout the structure, like here you can see they have more or less same size pores. This is dense membrane which is overall dense throughout the structure, and this is electrically charged throughout the structure. But when you look at an isotropic or asymmetric membrane, so they have different pore sizes, like in this membrane, and here you can see the top surface is like non-porous or dense, and then there is porous below that. And in this membrane there are some portion polymetrics, and then there are something liquid filled pores. So these are also asymmetric membranes. Before I start or talk about the membrane modules, here this is just one illustration for indicating that which kind of the or which type of the material are separated actually in different membranes. As we have talked that membranes are dense and as well as porous membranes. So overall when you look at the sizes of various molecules, so which molecules are separated where actually. So here if you look at some in conventional filtration, so that has pore size, some filter which are commonly used, so they have some pore size of 10 micrometer to 100 micrometer. Here this is the range of those, they are available in this range most of the time. So overall in this range you can just remove very large size molecule like starch, or sometimes somehow some parts of bacteria, but not even bacteria can't be removed because he has mentioned here 1 micrometer, so they can be removed with this. Yes, starch and all other molecules which are larger than that, they can be removed with this conventional filtration. Then when you jump towards microfiltration, so its size is more or less ranging from 1000 angstrom to 10 micrometer. So in this you can separate somehow bacteria. Yesterday you have seen their application was the separation of bacteria and yeast from some liquid stream. And then overall you can see there is the ultrafiltration, so that has a range of from 10 angstrom to 1000 angstrom. So overall this is used for separation of various components, like here you can see hemoglobin as well as these viruses can be separated by using these membrane. And then there was ultrafiltration, microfiltration, and there was nanofiltration actually here. If you remember, that has a size of 1 angstrom to the 10 angstrom. So overall reverse osmosis also lies somehow in the same range. So these small molecules which has small diameter like sucrose as well as sodium, water, they can all be separated in nanofiltration and reverse osmosis membrane. Actually when you look at various books, most of the time reverse osmosis membrane are referred as actually dense membrane which don't have any pores, so they can separate all the stuff. But nanofiltration membrane, the size was given that was like 1 angstrom up to 10 angstrom, so they can also be used for this removal of small size molecules. So from here what you can get the idea when you look at various filtration plants. Actually before going for, suppose you are cleaning the water and water contain various contaminants, various impurities. So before bringing your water to the final separating membrane, you can remove actually various components from the membrane by using different other membrane or filters. This will actually help to decrease the load of all pollutants on our last membrane. Then later when we talk about reverse osmosis in detail, we will talk about various issues or various problems which take place if you don't go for like these prior filtrations in membranes. But from here this is just to give you idea of average pore size of various membranes and relative sizes of solute. Like if you look at various salt, like there are salt, sucrose molecule, bacteria, viruses, water, so what are their molecule sizes. One thing here, maybe in the last slides you have seen the ultrafiltration, microfiltration and nanofiltration membrane pore sizes. Maybe they are not exactly here. Actually he has provided average pore sizes, so they can vary somehow in some text, so just don't get confused with this. So how does membrane work? When you look at these membranes now, how the separation takes place, you know that when we started with the membrane, so initially we said that actually membranes work somehow like this. Some feed enters in the membrane module. here this is membrane like, and something will pass through the membrane, so that's called permeate here, and something which does not pass through the membrane and leaves the system, that was called as retentate. So this was just simple illustration to show how membrane will make separation. But now we have to look at this, that how does membrane make separation. So here actually before we will just discuss these various types of membrane which are mentioned here, but just consider here it is hollow fiber membrane. There are the fibers small diameter, you can consider the fibers, and they are hollow from inside. You can just somehow consider them as a tubes, but there are also tubular modules and hollow fiber modules. So through these modules, if you look at this diagram at the bottom, just look at this, the feed is entering from one end, and overall if you have to understand this system, just consider it as a shell and tube diameter, shell and tube heat exchanger. So if you remember, there is some shell which is not actually shown here to you outside this, but in the above diagram you can see some shell, and inside there are the tubes. So there was something called tube sheet. So overall you can see here, this is also called tube sheet here in this even membrane. So these tubes are fixed to this tube sheet, and when this feed will enter through one side in this actually arrangement, feed is entering from one side. So when it pass through these tubes, actually tubes allow permeate to just leave. So permeate, some molecule of this feed will just leave through these various tube, and that leave as a permeate as shown here, while other which didn't pass through the membrane, that will move towards the other end of this tube, and that's called retentate, that will leave from this membrane module without passing through the membrane, without just crossing the membrane, or I can say without just permeating through the membrane. They are usually hollow and thin walled porous tubes. So hollow fiber also have some similar structures. Here a bit different illustration is provided, in which you can see that there is some membrane module. So inside this membrane module there are fiber, and they are in the form of U tubes. You can see that they are like U tube membrane modules. So hollow fibers here. Overall, some feed enters from this point, and when feed is entering from this point, some components of the feed can penetrate through the membrane or can pass through the membrane, and they will be collected inside in the inner side. Here they are entering from the outer side, and some can pass through this, some will not just pass through this membrane. So all those which are passed through this, they are collected inside membrane, and as you can see that here we have like membranes, they are hollow from inside. So all those liquid which is collected, or those component collected, are called permeate, and they are leaving from this end. While other definitely maybe you will be thinking how here some mixture feed can get mixed with permeate. No, you will have some tube sheet here which will stop the mixing of your feed with this permeate. While other component which can't penetrate through this membrane, which can't permeate through this membrane, so they will just leave towards opposite end and are rejected. As here I have shown you somehow one tube through this, some can penetrate through this membrane, can go through this membrane, can permeate through this membrane. So this is the permeate, and other which can't pass through this membrane will leave as a retentate. So here in this illustration you can see that some gas mixture is entering to the membrane module. This is somehow just similar like the system which you have seen here, tubular membrane module. So in this actually what is happening here, some of the component which just pass through these tubes, they are leaving, they are impurities, and while other which didn't pass through the membrane, they are just entering from one end and leaving on the other end. So they are retentate, and other which are just leaving out, they are called as which can cross the membrane and can go to the side, they are called as permeate. So now what are membrane module? This was about how membranes work actually on large scale. So on large scale, when we look for the application of membrane, so usually industrial scale membrane required large areas. So just recall something from the industrial example which we discussed last week, that was in the plant of benzene to synthesis of benzene from toluene. So there you remember that we have somehow 4 membrane modules or 4 parallel units were running, and I told you that each of that has somehow area of 4000 feet square, and overall required area was 16,000 feet square for that application. So how we can provide this much large area for the membrane in the plants? When you look at industrial plants, you need from 100 to 1000 square meter of the membrane area. So in order to provide that area, we have to place the membrane somehow in such a way that we get something economical and efficiently packed membrane area, because if you have to install membrane and you will need large area, then there will be problem to fix the equipment. And we have talked about this, that membrane have one property, they are compact as compared to other separation units. So the compact can be achieved actually if we just arrange the membrane in some specific pattern. So these packings or packed membrane are called actually modules. So when you look at membrane module, it means that how membranes are arranged on a large to provide large surface area for separation. So when we look at the membrane module, they are just categorized into various categories, like they are sometimes they are flat sheet membrane modules, they are spiral wound membrane modules, and tubular or hollow fiber membrane module, and one is other that is called capillary module, but we will just talk about these 3 types here in this lecture. So here just you can see the different membrane module are shown to you. These are spiral wound membrane module. The first one, I will just talk in detail about all this, how what are spiral wound module. The other is hollow fiber module. There are hollow fiber inside this, and this module is used as well. This is something also formed in the form of tubular structure, tubes are there, and this is in the form of somehow you can say honeycomb shaped structure. So they are also like one module, and overall here from this diagram you can see actually these kind of somehow filter. Actually inside those there are a lot of membrane, they are arranged for this separation, and they are installed in some plant. So let's talk about them briefly, each of these types. So first let's start with this plate and frame module. So plate and frame module are also called flat sheet module. When we will talk about the others, you will get this difference, that this is flat sheet module actually. So as its name indicate, there is some plate and frame. So overall these type of module contain, they have membrane, there are feed spacers, and then there are product spacers, and they are layered together between 2 end plates. So overall there are some frames or membrane modules, and they are placed or they are available between 2 end plates. So that's called plate and frame module. So here feed mixture, I will just talk about this, what he is saying, this feed spacer and product spacer. We will look at the diagram of this in next slide. So feed mixture is forced across the surface of the membrane. So when you are forcing the feed mixture, out of that some component will permeate through the membrane, and other components are rejected or leave the system as retentate. So mostly when you look at these plate and frame module in among various application of membrane which you have seen, so they are used for electrodialysis and for vaporation membrane application. So they mostly use this plate and frame module. So for small scale application, these are expensive. They are used for these small scale application, but they are expensive, as well as there are some leaks from gaskets. So when we have plates, and inside those plates we arrange all these membrane, and we have to give the path for feed as well as product to be collected. So some gaskets are used, and leakage occurs from those gaskets. So especially due to this problem, they are not used for gases, so they are used for electrodialysis and for vaporation. So this is one of plate and frame module shown to you. So overall just look, this is module in which you can see that some feed is entering. So just you can see the path of feed. So this he was calling as feed spacer. So feed spacer are provided so that feed can pass through, and inside there are like these membranes are arranged. So there are membrane module, they are placed in between these plates. So overall these are membranes, and when feed is passing through this area, is forced through this area, so you can see that membranes, these module are stopped from top and bottom. So any permeate which is collected can't leave, only it should leave through the specific path when you want to collect it. So when you will force the feed through this path, so some of the component which can permeate through this membrane, so they will just go or pass through this membrane module, and they are collected inside in this capillary, and overall this is finally collected as a permeate. While other which can't pass through this, they will just go through this path and collected in some area, and then they just leave as a concentrate, or you can call them that they leave as a retentate. Concentrate, actually he is giving this name because that's concentrated stuff. Some of those components are now in more concentration as compared to others, but this we have already studied, or we have given it the name of like retentate. So overall you can see that in half of the portion there is some stop disc. Stop disc mean this just separate two parts of this membrane module. So in first part just feed is moving towards top, and then in the second part overall you can see that some components of feed are extracted, you have removed them, but you bring back to them through another half portion, and try to extract more components, even to permeate some of the more components of this feed. So you have achieved maximum permeate which you want to collect or separate from here. So overall this is like plate and frame module. So I will just advise you, kindly just look at some video through YouTube, or just look at more images of this plate and frame module if still you have some confusion. Some of the other pictures are also provided in your textbook, so you can explore those as well. So from here you can see another arrangement. So in this feed is entering from the top, and in this like feed is just passing, or we should say there is one pass of the feed. Overall in this module feed is passing two times, or two pass type structure is here. But here this is just separated into two halves. One half passes through the one part, and other half is passing through the other part. So overall this is just passing through this, and finally collected. He has just shown feed in, and retentate, he didn't show here something called like permeate. So there will be definitely arrangement for permeate collection. So as this is some membrane module, so some of the component will be permeated through this membrane and collected as well. So this was first of the module, that was called plate and frame module. So there are the plates, and inside those plates we have the frame in the form of these membrane. They are separated with the spacer through which product can be collected as well as feed can be entered through the system. So next one, that is called hollow fiber module. So hollow fiber, something hollow which is empty, and fiber, you know that something which has very small diameter fibrous structure. So that's the hollow fiber module. In the first, which we have just seen, that they were flat plates. Actually you can see that your membrane are also like flat here. They are not like rolled, or they are not in the form of tubes, so they are flat sheets. But here when you look at these hollow fiber, they are tubular in the structure. Somehow there is the difference in the diameter of fiber and tube. Otherwise tubular module and hollow fiber module are they work or they are somehow structured in the same way. So hollow fiber membrane, they have advantage of like hollow fiber module, they have ability to be packed in a small area. Even large surface area membrane can be developed. Hollow fiber membrane is the ability, or has the ability, to pack a very large membrane area into single module. So here when we just arrange them in the form of tubular structure, so more surface area can be covered, or more surface area of the membrane can be provided even in a small packed module. So overall he has mentioned here like they have 10 times of spiral wound. Suppose we look at some arrangement, some structure in which if outer tube has the same area or outer structure has the same area, so inside that if you place hollow fiber they will have 10 times more area as compared to the spiral wound. We will talk about this, what is spiral wound. These hollow fiber modules are commonly used for reverse osmosis as well for gas separation and ultrafiltration. These are the applications in which they are commonly used. So here if you have we will again also look at its diagram in next slide. So if you will have high pressure, so in case of high pressure your feed should enter from the shell side. By the way, you have just seen one figure few slides back that was for hollow fiber, that green colored figure. Just look at this from here. Yeah, you can see that this one, if pressure is very high, so you can just introduce feed from this shell side. As I told you that you can just consider this hollow fiber and tubular membrane module just as shell and tube heat exchanger. So here in high pressure application your feed enters from shell side, while if pressure is low like up to 150 psi, so you just pass your feed through the bore side or tube side actually. So fiber size variation can lead to large variation in module performance. When you look at their size, actually fiber size, when fiber size will vary, so definitely there will be variation in module performance, because suppose fiber size is high, large, so it means you can just pack small number of fibers inside that module, and then in that case definitely surface area will be less. But if you just reduce their diameter overall, you can just pack more hollow fibers, and area required for separation will increase. By the way, just look for the reason why shell side is used for high pressure operation in this membranes. Just explore this kindly. So this is hollow fiber module. So overall you can see that there are some fibers actually, they are a bit different from tubes. As I told you that tubes will most of the time they have more diameter as compared to these fibers, and these fibers are small in diameter. Overall their structure is more or less same. So you can see here one of the hollow fiber membrane module. So this is from the website of any company which was provided these hollow fiber modules. So overall you can see that on the one end there is one hole, from here feed will enter, and when feed will enter, so the here feed is entering actually on shell side. So it will just keep passing through this structure. So overall those components which can just permeate through the these hollow fibers, so they will be just collected inside. So they will travel to the other end, as you have seen in the other slide. So here there will be leaving something called permeate. So others which can't pass through the membrane, they will be collecting on this end and they will leave as a retentate. So definitely here will be at the end of this membrane module, after this nozzle, there will be something called tube sheet that will be just separating this retentate from permeate. Because if you just don't install any tube sheet on this end, so this permeate and retentate will be again mixed. But there is a tube sheet which just separate this the retentate or concentrate from permeate, which is collected here. On the other hand, here you can see in the this diagram overall, this is quite similar to the shell and tube heat exchanger, in which you have like two inlet and two outlets. So overall you can see on this end these two ends are provided, and there are tube sheets on both ends. And here if feed enters, suppose from this side or from this port, from bottom port feed is entering, so at the end some portion of the feed is penetrated through the membrane, that permeate through the membrane and is collected as a permeate, while other which can't pass through the membrane, that will just leave from here as a retentate. So there are various actually structures, or they can be arranged or they can be placed in the form of various structure. Like here you have overall two opening on shell side, and I should say just to give it similarity to shell and tube. So there is shell side two opening and two tube side, but in this on shell side there are two, on tube side there is only one. By the way, here you will be thinking, so on the other end either tubes are open? No, they will be just clamped on the one end. On this end they are definitely stopped. They don't have any opening on this end, but they are just stopped. They will be closed on this end. So anything which goes through or which penetrates in these fibers, so that will be collected on the other end. On this end nothing will happen. But in this kind of module, so definitely you can collect product from both ends, or maybe you can stop one of the end and can collect on the other end. And sometime this is also provided something you remember, that was called sweeping in the start lecture when we were looking at the membrane separations. When we started with this, there was something called sweeping gas, or sweeping is used for pushing off permeate. So sometime you can use also this. Suppose you want to collect your product of permeate from this side, so from this end you can just introduce sweeping gas. So based on your application or based on your requirement, you can modify the these modules. So then there are spiral wound modules. Spiral wound, as its name shows, spiral, you know that something which is rolled. Our wound is something folded. So spiral, something which is folded in the form of circle. So that's called, like suppose if I just fold something in the form of circle, so that's called spiral wound module. So let's look at them. So here membrane envelope of a spacer and membrane wound around a perforated central tube. There is some perforated tube inside these modules. So around that actually you just fold in a circular way. There are spacers and membrane, they are folded around that tube. So module is placed inside a tubular pressure vessel. Overall around these main modules there is some vessel that can bear high pressure. And most of the time when you look at spiral wound module, so they are used for reverse osmosis and gas separation application, and they are available in the size of like 2 by 3 feet diameter of that module, and usually they are 3.5 feet long. So let's look at these figures to get more clear idea about these modules. So just at first diagram, from here you can see that what we have, actually we have membrane. There are some membrane, and then there are spacers. As you have seen, this terminology was provided for flat sheet membrane. So when you have looked at plate and frame membrane, so when you have seen plate and frame membrane, so there you have seen that there were product spacers, as well as then there were like something called feed spacers. So same, if they are provided and they are not in the form of flat sheet, what we do, we just spiral wound them, we just fold them in the circular shape, and here you can see that they are just membranes are folded. So for example, you just take a page and fold it, and inside that page you just place your you can place pen. So overall this pen is something perforated tube here. So when something will pass through this membrane, so permeate is collected inside that membrane tube, and that will pass through the membrane and is collected inside this tube, and will leave from this. Anything which did not pass through the membrane is collected as a retentate. Here this module is provided, as he has mentioned, that they are cased, or they are packed inside some packing which can bear high pressure, because as we have seen they are used for either something gas permeation or reverse osmosis. So for those applications usually high pressure is needed, so the casing which you are using should be able to bear high pressure. So overall in this module you can see that feed is entering from one end, and they are spiral wound here module. some part will just permeate through the membrane, and they are collected inside inner tube, central tube, and which are not collected, they leave at the other end as a retentate. So maybe this is not clear to you, but one thing I wanna just tell you here is just one video I will upload for with the name of this spiral wound module. So just go through that video. That's 4 to 5 minute video, and that has explained properly that how they work. If you are interested to look how they work, or how the main retentate and permeate are collected through these membranes, so just go through those that video, and you will get better idea about it. So then there are here this slide actually shows, now we have seen various module, and suppose now you are thinking which module we have to use. So this is a brief comparison between these modules. So no need to remember this is just for your information. Also it is not from your textbook, from some other book. I have just collected this. So just to give you idea, first point here is for manufacturing cost. So you can see that here US dollar per meter square are provided. So hollow fiber, capillary fiber, there are spiral wound, there are plate and frame, and then there are tubular at the end. So out of this, as we said, we didn't discuss the capillary fiber. Actually capillary fiber are somehow similar to hollow fiber, just they have very small size. There are capillaries as compared to these tubes and fibers. But overall you can see that their prices are given here per meter square in US dollars. So most of the time plate and frame and tubular are the expensive one, but other just leave second point. Maybe if you see at this permeate side pressure drop. So permeate side pressure drop, if we look at the pressure drop, so this is high in hollow fiber, but it is moderate in other two, and the last two, plate and frame and tubular, it is low pressure drop. So these are the various parameters which you have to look for when you are going to select the different modules for industrial application. So more than one parameter are looked, and then you decide that which should be used. Like as he mentioned last point, like limited to specific type of membrane material. So hollow fiber and capillary, they are used for special type of material, but others they don't have this limitation. So any membrane material you can construct spiral wound, plate and frame and tubular, but hollow fiber and capillary, they are used for some specific materials. So that's it. Up to here actually we have talked about membrane modules. So just briefly summarize this, that membrane modules are actually you can simply say that that's the arrangement of large surface area membranes for industrial application, and there are different module types. They are simply flat plate, there are tubular, and they are hollow fiber, as well as there are spiral wound module. So we have just talked about them briefly about each type. We didn't go in much detail. We have just talked how they are formed or how they are arranged, actually how they look like, and which special application they are usually used. So then there is next and the last topic of this main lecture, that's the membrane synthesis. So how now these membranes are synthesized? Up to here we have seen that what membranes are, what materials are used to synthesize the membrane, so like polymer and inorganic, and then we said that when you have to use them on large scale, so how they will be arranged. We have talked about all this stuff. Now if we have to install that membrane and we have selected one of the types, so how to synthesize that membrane. So several different techniques are available to synthesize these membranes. So to prepare this synthetic membrane or man-made membrane. So some of these techniques can be used to prepare polymeric as well as inorganic membrane. So here I have just given you the names of those techniques because this is not part of our this syllabus to talk about in detail. So that's that will become are that's available for higher level for graduate course, but just you should remember few names that how they can be synthesized. So some technique involve actually phase inversion technique, there is interfacial polymerization technique, sintering, stretching, there is track etching and sol gel processes, vapor deposition, and finally solution coating. So all of these technique involve a lot of details, and you don't need to go in detail for this subject. At least if you are just interested, you can go through some texts in which they are explained. So overall, if I just summarize here these membranes, so if you look at this membrane material, their preparation, structure, synthesis. So first of all, you can see, you can start from this point, membrane material. As you have seen that membrane material were commonly, I told you yesterday that they can be just separated into two, like organic polymeric membrane and inorganic materials membrane or inorganic membrane. So they are usually oxides, because you remember there was aluminum oxide, silica oxide, zirconia and such stuff. They are ceramics, metals, they are formed of these material. And then there are mixed matrix membrane and mixed matrix membrane or composite membrane. Composite mean that's formed of more than one component. Sometimes they can be organic and inorganic, they can be mixed, and sometimes mixed matrix involve like one membrane which we have seen, that was something polymer matrix and some liquid inside that. So they are also called like this mixed matrix membrane. So commonly two categories which we have discussed here, they were organic and inorganic. So this third one, just remember that there are mixed matrix or composite. They are formed of two, mean like they are not pure organic, not pure inorganic, they will be just mix of these two. So after that, you can see that when you look at membrane cross section, so they are the one diagram which we have discussed, isotropic or symmetric. So they were like anisotropic or asymmetric, in which their structure was not same throughout like the membrane, and then they can have like thin layer or mixed matrix membrane, bi or multi-layer. You have seen in various slides some of these types. So after this, their shape, how they are moduled. So they are in the form of flat sheet, hollow fiber, capillary or tubular. So we didn't talk about capillary, but capillary, they will be from this name capillary, small size tubes. They will be less than this hollow fiber, and tubular less in diameter. So then the finally membrane preparation or synthesis methods, which in the last slide they were just names were given, same names are provided here. So that's it. Up to here in these main lecture series of this week, we have discussed about membrane materials. I have just summarized in the last slide. Then there are membrane types, so dense membrane and porous membranes. So and in porous there are microfiltration, ultra and nanofiltration membrane. Then there are membrane modules, so plate and frame. There is spiral wound and hollow fiber or tubular module membranes, and then their synthesis involve various techniques. So that's it. This most of the stuff is from your textbook, but few things I have brought from other book, like one book mentioned here. So if you further want to read, someone is interested in reading about these membranes, so this book is dedicated for membrane. So membrane technology and application by Richard Baker. So this guy has written our work a lot on the membrane. So this is one dedicated book for membrane, and some of the part of this lecture is actually from this book, but most of the part is from your textbook. So you can just go through your textbook and you will get the idea. Other which is not available in your textbook, just go through slides, lecture, and maybe you can consult this book. Thank you so much, and kindly, if you have any question, just post by email, or you can drop a text, and try to finish all these lecture by Saturday of this week. Thank you.